Let's talk a little bit about bulk transport. I've actually witnessed this. I've experienced this myself, being shoved onto a train in South America and also in Central America, where there were so many people that there was no space, and there was actually people pushing others onto the train. It was ridiculous. A friend of mine went to China and said that this actually is not a rare occurrence. They actually have porters that will actually push to try to get as much bulk inside the transport vehicle as possible. This doesn't look like it's that crowded in here, so I don't know what this lady was doing. But when we talk about bulk transport with respect to cell membranes, we're talking about big amounts of stuff getting in or getting out. A lot of times you can get these sucrose molecules in through a co-transporter, but it's not efficient enough to do it one at a time. The cell's has evolved to have processes in place where tons of stuff can get out and tons of stuff can get in at once. This is called exocytosis and endocytosis. Do you think this takes ATP? Do you think it's an active process? The answer is yes on both of those. So make sure that you look out for the ability to be able to explain how this happens, and then distinguish between exocytosis and receptor-mediated endocytosis. First, we're going to talk about large molecules crossing the membrane via vesicles in this bulk flow. I've said earlier that it requires energy. Here's a vesicle of stuff that's either getting ready to leave the cell or it has already entered the cell. But first we'll talk about exiting or exocytosis, the way that stuff gets out of the cell. And here you can see that there's a vesicle of phospholipids, that fat coating around something that the cell wants to get rid of. Maybe this came from the Golgi apparatus. Maybe it's a modified amino acid chain, and it's going to then bind with the phospholipid bilayer of the membrane and be released. And this is how the cell gets rid of contents very often. Here's the picture of the Golgi apparatus. Remember that there's a receiving side and a shipping side, and then you can see down here the item meshing with the plasma membrane and then leaving. Many secretory cells use exocytosis to get their products outside of the cell. Endocytosis, I always think of IN or in. It's bringing in something. The cell takes in macromolecules by actually forming vesicles from the plasma membrane. And you can see this here. The plasma membrane is actually encircling the item, and it's creating a pouch there to bring it in. Yes, it takes energy. It's the reversal of exocytosis. It involves definitely different proteins for this process to take place. There's three different types of endocytosis. There's phago, pino, and receptor-mediated endocytosis. You can see up here phagocytosis has been, if you want to simplify the Latin phrase, it's to eat, and pino means to drink. So I don't like this at all. The drinking one, we've already talked about aquaporins. That's how water gets into the cell. Maybe it's drinking the kind of environment that's around it that has both solutes and solvents and maybe even living things in it, if it's a bacteria or a white blood cell. So here's phagocytosis. This is how some food or other particle can get into a cell. Here you can see an amoeba, which is a single-celled eukaryotic organism, and it is actually digesting a bacteria, and it's doing this by phagocytosis. This vacuole here, this food vacuole, fuses with a lysosome to digest in this case, a bacteria, and that's how the amoeba will get some nutrition.
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
You just saw an example of pinocytosis. Basically, it's when the environment is brought in. It's an aqueous or liquid environment. You can see it here. It may have some solutes in it. Here is an example on the right side. And then finally, receptor-mediated endocytosis is when a ligand or something that causes an activity to occur in a cell when a cell protein has been bound from the outside, binds to one of these receptors, then the cell can actually pinch in and grab the items that are out here. Maybe these green things are what it's focusing on trying to get inside. And it's these purple guys, these ligands, which will then stimulate that deepening of that pit in order for the pinching off of the phospholipid bilayer. You can see it happening here on the left. The pit is deepening and then eventually it will pinch off. Ligand is any molecule that actually specifically binds to a receptor and causes something to happen. Make sure you spend a few moments answering these objectives, and I'll see you tomorrow. Thanks.